Now let me turn to this evening's presentation. Children under stress, vulnerability and resiliency in the developing brain. Now, psychiatrists and psychologists have long recognized the adverse effects that excessive and unavoidable stress can have on young people's cognitive and emotional development. New studies of the brain, the nervous system, and the neuroendocrine pathways are providing clinically relevant information about the connections between stress and behavior in pre-adulthood. And today's speakers will help us to understand the mysteries, and indeed they are mysteries, of the young brain. How it grows, how it develops, how it changes during adolescence. In particular, we're going to hear about how stress can affect the brain and how that relates to intellectual and emotional development, as well as to behavior. Our first speaker is Dr. Bruce McEwen, Rockefeller's Alfred E. Mirsky Professor and head of the Harold and Margaret Millikan Hatch Laboratory of Neuroendocrinology. Bruce's groundbreaking work explores how stress and hormones affect the brain's structure and function. Dr. McEwen will be followed by Dr. B.J. CASEY, the director of the Sackler Institute for Developmental Psychobiology at Weill Cornell Medical College, just across the road, where she is the Sackler Professor of Psychology in Psychiatry, Neurology, and Neuroscience. Dr. CASEY is a pioneer in the use of neuroimaging to study brain development in children, and we're delighted that she is here with us tonight to share the latest insights from her research. You will have more information in your program for details about Drs. McEwen and CASEY. After we hear from our two speakers, we will then open the program for questions and answers. So we'll all go around this table, and you can ask your questions then at the end of both talks. So first of all, let me introduce Bruce McEwen. Could you please come to the podium, Bruce?
Thank you very much, Paul, and good evening. I'm going to be talking to you about stress. And stress is something that I think we all know about in our daily lives. In the course of this, and as Paul Nurse said, I'll be focusing on the role of the brain and its response to stress. And in the course of this, B.J. CASEY and I will each talk about respective parts of a collaborative study that we've been doing together with an MD PhD student, Connor Liston. But first, let's get into stress. Now, this is one view of stress. The way that I think I prefer thinking about it, we know that there's something called good stress or positive stress, where we can feel an exhilaration from a challenge that has a satisfying outcome, with a sense of mastery and control and good self-esteem. And then, of course, bad things do happen, and there's tolerable stress or adverse life events, but we have good social and emotional support. And then there's something which we can call toxic stress, which involves experiencing chaos, perhaps having experienced abuse, neglect as children, or in adult life, and above all, the absence of good social and emotional support. Towards the end, I'll talk about something, a study called Adverse Childhood Experiences, or ACE, resulting from toxic stress. And actually, just yesterday, I was at the Center for Disease Control in Atlanta, which has done extensive studies in this area, and I'll refer to it at the end. And in the adverse childhood experiences, the ACE, the consequence of this for people, there are long-lasting cognitive, behavioral, and systemic effects on the body and brain. We'll come back to that at the end. Now, when we think about stress, and I'm a neuroendocrinologist, so I think both about the brain and about the hormones and the rest of the body, we think of cortisol. Cortisol is a hormone produced from the adrenal cortex. It's released when a hormone called ACTH is released from the pituitary gland. The ACTH is released when Two hormones come from the hypothalamus in the base of the brain. One is called vasopressin and the other is called CRH, or corticotropin-releasing hormone. These hormones are released from nerve cells, cause the pituitary to release ACTH, and that causes the body to produce cortisol. Now, what causes the hypothalamus to release these two hormones are neural inputs from different parts of the brain that are activated by experiences that are stressful to us. Now, one of the things that we have to remember is that Mother Nature put cortisol there to do good things, so that in the acute aftermath of stress, we couldn't actually live very well without our adrenal glands. Cortisol does some very beneficial things. It enhances our immune system. It improves certain aspects of memory. It helps with energy replenishment after something stressful happens, and it improves our cardiovascular function. But when cortisol is present in excess or is secreted for long periods of time in chronic stress, then it can suppress the immune system. It can impair memory, and I'll say a little bit more about this. It can promote things like the loss of calcium from bone, the wasting of muscle, and contribute something called the metabolic syndrome. So we often describe this as an inverted U-shaped dose-response curve. A little bit of cortisol is very important if it's properly regulated, but too much can cause problems. Now, the brain has a dual role of controlling the stress response and also controlling our behavior. Three areas of the brain are particularly important, and both BJ CASEY and I will be talking about these areas. One is called the prefrontal cortex. The other is called the hippocampus. And the third is called the amygdala. The prefrontal cortex has an important role as a top-down control of the stress response. It helps to balance it. On the other hand, the amygdala, which has to do with fear and emotion, turns on the stress response when something happens. And the hippocampus is also, like the prefrontal cortex, involved in shutting off the stress response. Well, while it's doing this, these same brain regions are also involved in emotions and cognitive function. The amygdala is the seat of fear and anxiety and strong emotions, and also the seat of aggression. The hippocampus is important for remembering events of daily life, negotiating space, remembering where we are and what we are doing when something very powerful happens to us. It's a very important memory organ, and the prefrontal cortex has a role in decision-making, in working memory, and top-down control of impulsive behavior. And when these brain regions malfunction, then you have psychopathology, as well as alterations in our stress responses. Now, what we do in our laboratory is to study the cellular and molecular mechanisms by which the brain actually changes during stressful experiences. And what I'm showing here this is a nerve cell, and here is the genetic material is in the nucleus, the DNA is here. These are called dendrites. And these dendrites receive inputs from other nerve cells on these spines that actually represent synaptic contacts. And these synaptic contacts are places where chemical neurotransmitters are released to activate from one cell to the other and create the activity that constitutes how the brain works. There are cells in certain parts of the brain, particularly this area called the hippocampus, that continue to be replaced during adult life, the process called neurogenesis. And this seems to play a very important role both in emotional and cognitive function. And the most important thing that we and others have discovered is that the adult brain is structurally plastic. It's capable of changing both in terms of the number of neurons being replaced in the hippocampus and the number of synapses and even the length and branching of these dendrites. So that what we know, particularly for the hippocampus, is that the hippocampus is a brain region that shrinks when an animal, or some evidence that now for people, when there is long-term stress. The nerve cells actually shrink and there are fewer new neurons produced in the dentate gyrus. It also is affected by diabetes, and it also shrinks and begins to malfunction in people with type 2 diabetes. With long-term depressive illness, it becomes smaller and it's impaired in function. And antidepressant drugs turn out to increase especially the production of new nerve cells and help to reverse this shrinkage seen in depression. People with low self-esteem, a very relatively new finding that needs to be replicated, but there are pretty good suggestions that low self-esteem involves having a smaller hippocampus. And one of the interesting challenges is if we could improve someone's self-esteem, could we enlarge their hippocampus? So that's some of the things we know about the hippocampus. The other brain area that, well, as I said, the neurons in the hippocampus shrink with repeated stress in animal models and some evidence now in people. Neurons in the prefrontal cortex also shrink with repeated stress. And this is a story I'm about to tell you, but I want to say that while chronic stress causes neurons to shrink and these dendrites to become shorter and less branched in the hippocampus and prefrontal cortex, it causes neurons to become increased, the branches to become increased in the amygdala. And what happens when you have chronic stress and the animal amygdala is enlarged is that the animal becomes more anxious and more aggressive, while the cognitive functions that are supported by the prefrontal cortex and hippocampus are impaired. Now, I want to tell you briefly about an experiment which was carried out by Connor Liston working together with Dr. CASEY and myself. The first part, which I'll tell you, she'll tell you the second part, involves looking at the prefrontal cortex. Now, this is a neuron in the prefrontal cortex. It's filled with a fluorescent dye. You can see the dendrites. And here at high power, you can see the spines, the synapses that represent these chemical connections between nerve cells. And what we've found over a number of studies is that repeated stress causes these dendrites to decrease in branching and reduces the number of synaptic connections. So there's less information then coming into these nerve cells as a result of chronic stress. If you, as another group in England did, if you lesion the prefrontal cortex, that is actually destroy parts of it, and look at the ability of an animal to conduct a number of cognitive tasks, make a discrimination between a number of stimuli that it needs to use to find a food reward, or doing a simple reversal of what cue predicts where the food will be, the lesion doesn't have any particular effect. But if you ask the animal to completely reorganize to use a new set of cues, this is called an attention shift, then the lesion of the medial prefrontal cortex impairs the animal's behavior. Well, what Connor Liston found is that repeated stress causes almost as large impairment of this attention shift without impairing these other somewhat simpler tasks. The difference between the lesion and the effect of stress is the effect of stress is reversible. If you stop the stress in the animal over a period of several weeks, the brain and the behavior return to normal. And you'll hear more about this in relation to what happens with stress in medical students when Dr. CASEY talks. But I want to finish up and go back to this slide, in which I've, of course, emphasized the important role of the brain, both in the control of the endocrine hormonal response to stress and in terms of behavior. And I've emphasized areas of the brain like the amygdala, the hippocampus, and the prefrontal cortex that are very important in emotions, in decisions, and in memory, and also the importance of the brain in maintaining good self-esteem. Now, if we come back to the topic of toxic stress, I said at the beginning that there are long-term cognitive and mental health and physical consequences of a child being abused and neglected, which last into adult life. And having just been at the Center for Disease Control, this is actually a figure from their website that reflects a series of studies that have been going on for more than a decade, headed by scientists at the Center for Disease Control and collaborators at other universities. And what you see here is at the bottom of this pyramid, you have the adverse childhood experiences. You then have social, emotional, and cognitive impairment that results from growing up in a chaotic and abusive environment in which there is a lack of adequate social parental love and support. The adoption of health risk behaviors, things that relate to substance abuse and many other mental disorders, including depression, vulnerability to post-traumatic stress disorder. And then what's less appreciated, it's not just the brain. There are also consequences in terms of increased frequency of cardiovascular disease, of diabetes, and earlier death. That is, mortality, the life expectancy is reduced in people who've had these adverse childhood experiences. And of course, one of the big challenges for our society is how to deal with this effectively. Yet, fortunately, most children grow up in much more supportive environments, and yet there are components of social and emotional support that are, of course, very important, and we're going to be hearing more about those from Dr. CASEY. So with this, I will then go on to Dr. CASEY. Let me just, thank you.
It's a pleasure to be here with my distinguished colleague and collaborator, Bruce McEwen, but even more so with parents and grandparents, as I'm a parent of a teenager myself. So this is a very important topic to me. So what I'd like to do is to share with you some studies about the adolescent brain that we've been involved in at the Sackler Institute. But before I even begin in talking about this research, I'd like to share some health statistics with you to set the stage for the importance of these studies. It's the case that by the time our children leave high school, a third of them will have had a major depressive episode. It's also the case that the 15,000 adolescent deaths each year in the United States, a third of them will have been due to suicide. I think that these alarming statistics really underscore the importance of us understanding what is happening in the adolescent brain. Now, there are 3 points that I'd like for you to sort of take home from this in terms of changes that are going on in the brain and why adolescence is such a period of vulnerability, and also why some individuals may be more susceptible than others. So the first is just to recognize that different brain regions are developing at different points in time, and this influences behavior, particularly during adolescence. And I'll give you examples of this. A second is that there are individual differences in the development of these regions, and much of this is driven by biological predispositions or genetics, which we're not going to talk about today in the interest of time, but maybe during Q&A. And then a third is, and this will underscore and bring back the important work of Bruce McEwen in terms of vulnerabilities during adolescence for disorders of anxiety and depression are actually exacerbated by chronic and long periods of stress. So the overarching question is, what's changing in the brain during adolescence. So we see what many of us in looking at our children think is aberrant behavior. And what I'd like to first sort of provide for you are arguments that a number of scientists have made in Time Magazine, Nature, a number of publications recently, and that is that prefrontal cortex that Bruce mentioned before, that's so important in regulating our behavior and emotions, that this region has a very protracted course of development, and so it's very slow to develop. Now, I'd like to just ask the question to you. If it's all about the prefrontal cortex developing, then why aren't we seeing children engage in these aberrant behaviors? They would be at least as bad, if not worse. So the research in our laboratory is really trying to understand how is this region interacting with other systems that are key for emotional processing and emotional reactivity. And so in this little simple cartoon, you can see that there's across age, and this is right at the period of adolescence. As this brain region is developing, the prefrontal cortex, we're seeing that the functional development of the amygdala is occurring sooner. So in children, there's not competition between these two regions because they're both still developing. In adulthood, these regions have matured. But during adolescence, you get the system that's just banging, the system that's so important in picking up on cues in the environment that signal safety and danger. And we all know, adolescents, many of them, in any sort of neutral situation, they can read emotion into it. So this is the system that's hopping during that time. So now I just want to show you examples of some of the research where we've tried to investigate this using brain imaging. I've already had some questions about healthy volunteers. The studies I'm talking about for the most part today are healthy volunteers who come in. This is a non-invasive procedure, and maybe during Q&A we can talk more about it. We find that if participants are calling us to try to get in another study, that's a good sign that it's a child-friendly environment and research program. The way that you look at brain activity in these scanners is to present stimuli that the children have to respond to, and then you can look at what parts of the brain are changing as you present these different stimuli to them. In this study, we had emotional faces that were either fearful, where you can see a few, or some of them that were calm, neutral, if a face ever is neutral, particularly to an adolescent. And now what I want to show you is how the activity in this amygdala system that's so involved in processing emotion, how that activity changes from childhood to adolescence to adulthood. What you see is, this is just taking a picture in the brain as if you were looking straight at it, about midway. And this is the amygdala that Bruce mentioned. These two hot spots are where it's active when they were viewing these fearful expressions, and this is activity from childhood to adulthood. The blue are children. See that elevated initial activity in the adolescents, and then adults it comes back down. But the question is, in trying to understand who may be a vulnerable teenager and who may not, teenagers are going to respond. There is going to be some heightened emotion. But what's key is when they can regulate it, right? Then you got to bring it back in. Get over yourself. Calm down. So let me show you how we look at that as well with imaging. Actually, my son says, "Get over yourself" to me all the time. Like, parents are just there watching sometimes. So this is showing you the same data from the previous slide, but these are early trials. So you see it's very high. And then in late trials in the experiment, really what you're doing, we're presenting empty threat. They're fearful faces, but nothing's happening to the volunteer. So they should eventually just sort of habituate, like same old, same old. And so you see a number of individuals actually come down in the degree of activity. We've actually reported in earlier work that for some reason teenage girls have a more difficult time in habituating with repeated exposure to threat. Now, we wanted to sort of understand why some individuals just really couldn't regulate this emotional response. And so we had them answer questions about everyday anxiety. And I just wanted to throw up a few examples. When I get finished, frightened, I feel like I'm choking. I get really frightened for no reason at all. So these are just questions they answer about themselves. And then what we observed is if we took that overall rating, that score, and tried to relate that to the amount of activity that was still going on in this emotion region by the end of the experiment, we got a nice association. So basically, if the child rated themselves as very anxious, they still had high activity by the end of the experiment. But what's more important is, how do you regulate the system? And so what we looked at were brain regions that were connected to this region and were tightly coupled. And that brain region is the prefrontal cortex. And in this slide, you see that those individuals who didn't have very much activity in the prefrontal cortex, this is the front part of the brain, those were the ones who weren't able to bring that activity down. Now, I think pictures are worth 1,000 words, particularly in brain imaging, so I just want to show you a movie. The top is an example of anxious individuals. Here's the bilateral activation when they're presented with a fearful expression. And what you see is it's maintained, even with repeated exposures. And then I'd like for you to compare that to a less anxious individual and see its bilateral activity. But I want you to see how it begins to diminish, so much so that it ends to almost go back to baseline and turns blue. Now, this is really quite interesting because these were typically developing children and adolescents. In our previous work, what we showed with teens with anxiety and depression is that this area, the amygdala, was activated, and not only that, that the degree of activity in this region was correlated with the number of anxiety symptoms leading to its diagnosis. So take-home point number one is that during adolescence, there's this imbalance between these brain regions where the emotional system is really driving things. So in the heat of the moment, when a child has to make a decision or is feeling a certain way, it's going to win out. It's also the case that there's individual variability that we saw in terms of how well teenagers are able to, and individuals in general, are able to regulate that emotional response. So now I want to briefly mention two studies where we bring back in the importance of considering stress and its influence on these brain systems. And the first study is to talk about the effects of early postnatal stress and infancy and early childhood and how that plays out later in development. And then the second is to look at the effects of stress during later development. So our example, or the variable that we're using to sort of examine postnatal stress, is working with a large cohort of families with children who have been adopted from orphanages abroad. We feel like that time in the orphanage when there wasn't one-on-one direct care with a parent was a stressful event. It's not the same as the manipulations that we do in the rodent, but there are a number of factors that come into play. None of our children were tested until they were at least in the country for 2 years, because we wanted them to acclimate to the country as well as to their families and novelties. Some of them had never even experienced texture in foods. And the children came over, were adopted anywhere between 6 months of age all the way up to 60 months of age. And we're continuing to increase the cohort. It's currently 70 families who have been wonderful in participating in this study. What we've been doing with this work is trying to, there's no way that you can control for the conditions that these children experienced. Some of the orphanages are quite good at providing resources to the children, and others are really bad. Some of the children, they arrive with rickets and a number of complications that we just don't typically see in the States. But we wanted to use the stress model that Bruce had developed to look at what are the long-term effects of this stress on the developing brain and brain structures that we know are impaired and impacted by stress in the animal models. And what I'm showing you now is that hippocampus and the amygdala regions, two regions that Bruce talked about before, Remember, the hippocampus is trying to shut stress off, but we see in our children who have been adopted that they have a diminished volume. The size of the structure is smaller. But the amygdala, the system that's just jamming about emotional information and trying to interpret it, it's actually larger. Now, these measures are based when you control for overall brain region, so these are regional specific changes long after the stressor has occurred. I should also note that many of these children come in with diagnosis of anxiety disorder. Now, this is interesting in light of some of our other work where we looked at children with generalized anxiety, and we looked at the structure of the amygdala, and we actually saw these are typically developing children, and these are children who have been diagnosed with generalized anxiety disorder, and so they have a larger amygdala volume as well. So take-home point 2 is that the imbalance between these two brain regions, the amygdala and the prefrontal cortex, during adolescence may be exacerbated by stress that occurred many, many years earlier. So we have to be aware of that in terms of really listening to teenagers and knowing about their history when they come into the clinic. And I just sort of have a cartoon here where you see that that activity is elevated. The last study I want to mention briefly because Bruce really set the stage for me, and this is a collaborative project with Bruce McEwen, myself, and Connor Liston, where we looked at the effects of stress on prefrontal function in medical students studying for the boards. This is a very stressful situation, and one of the functions of the prefrontal cortex is allow you to flexibly shift your attention. Now, I'd like to argue that when you're studying for the boards, you don't want to be shifting your attention around to what your peers are doing. Sometimes it's very good to focus in on the stressor. But this can also result in ruminations in other situations. But this shows you that attention shifting, it took much longer for medical students who rated themselves as very stressful during this time of studying for the boards. Now, let me give you some examples of questions that they are asked on the perceived stress scale that Connor collected. Some of them include, how often have you felt nervous and stressed? You also ask questions on the flip side of how often have you felt that things were going your way, just to make sure that you're getting an accurate reflection of how that individual is feeling. So those individuals who were having difficulty, though, were those who had the highest perceived stress score. Now, how does that play out, that not being able to shift attention as well in these brain systems, particularly the prefrontal cortex, which is supporting this function? Whoops, I'm going to come back to this one. So if you look at activity, this is low stress, individuals who didn't report much stress during this period. This is high stress, and this is the degree of activity in the prefrontal region. So you're seeing lower activity in this region in the high stress group. Now I want to bring it back and integrate the human and rodent work. This is the animal data, the red and the green. This is stress. This is the control condition. This is stress, and this is low stress. So it really mimics beautifully, but in the human, we don't have as fine of analysis of the neuroanatomy that we do in these animal models. So this collaboration was just a beautiful one, and we could also actually link it to everyday stress that we experience and how the brain can be reorganized by that, whether or not it's adaptive or not. Now, it's also very important for you not to leave here thinking that medical students are brain damaged. So Connor had them come back in after they'd taken the boards and they'd had a vacation. And this just shows you that after they've taken the boards, you see that the attention shifting, they get better, they're faster. It's also the case that their stress comes down. And the beauty of this is that in the prefrontal cortex, you actually see that the connectivity to this region actually improves, increases. So the final take-home point is that the imbalance between these two brain regions, the amygdala and the prefrontal control regions during adolescence, may be exacerbated during adolescence, particularly when there are long periods of stress. And we know our teenagers, when they're studying for and preparing for SATs, even college students getting ready for GREs and the boards, these are all periods that we really need to be aware and at least in the background, monitoring our children to make sure they're doing okay. They're going to have their emotional burst, but usually they quiet down. It's when they don't that we become concerned. So just to conclude, again, just understanding how the brain is changing during this period gives us insight in terms of some of this adolescent behavior that helps kind of understand why they do some of the things that they do. And to recognize that there are individual differences. There are a number of adolescents who come through this period beautifully. So there's resilience as well as vulnerability. But those individuals who are most vulnerable, we really do need to be aware of the effects of stress on exacerbating that vulnerability. So I'll stop there. Thanks.
Thank you. Thank you, Dr. Casey. We're going to go over here and take questions and answers. Yep.
Great talk. Thank you very much, both. Bruce, I wanted to ask you if there is some look comparing the oxytocin and calm connect response in adolescence compared to the fight-flight response, and particularly in differences between male and female.
So far not. What, of course, you bring up is a very important hormone, another hormone besides cortisol called oxytocin, and there's yet another one called prolactin. And there's evidence mostly from experiments in animal models that both of these hormones, which are also elevated during stress, actually have anti-stress effects and anxiolytic effects. And since these hormones are produced in somewhat larger amounts in females, especially during different times of reproductive cycle, one might suspect that women are more sensitive to this. And I think in animal models we know that this is the case, but so far this hasn't been translated into humans, and it's a very interesting and important topic.
I think there was another question near the front. Yeah.
Very good talk. I enjoyed it. In the comparing this stress, the non-stressed adopted infants, showing that the non-stressed, I guess, had larger. Who are your controls? Are they matched for ethnicity and age, and because both those children looked Oriental or Asian?
So that's a very important question. There is no appropriate control group for these children where we have very limited information about the backgrounds and the orphanage. Basically, the research, in most of our research, we compare them to one another, and we use the age in which they were adopted and the amount of time spent in the orphanage to look at how these changes are occurring. And there is an association between length of time in institution and the size of this amygdala region. And the hippocampus, not so much. And we think that may be due to, well, there are other things that we see with these children. They tend to catch up very quickly in terms of cognitive abilities, but it's not that everything triggers some of these children. It's when they are triggered, it's very hard to get them to settle. Now, I say that, but there's so much individual variability. We've begun to do some genetic studies, particularly of polymorphisms, where they're very common in the Asian population, because as you can imagine, the majority of our sample are Asian girls, and they're doing quite well, and it's just lovely to work with these families.
Right, there's a question here just to the left. Could you wave? Yeah. Great.
Thank you very much. A question and a comment. First, thank you to Rockefeller University for inviting parents to this evening. We're very, very appreciative. The comment was that when I saw the cartoon about the amygdala exceeding the performance of the prefrontal cortex, I thought, why on earth did we make 18 years old legal adults? That's a political comment. But the question was, did you study for genetics? I see that you studied for stressors, which are external, but did you also study a genetic component?
So for human genetics, any associations take extremely large sample sizes. And so really we're talking about thousands that we would need, which is why we're trying to ground our studies in mouse models where we can look at mice that actually mimic the human polymorphism or a biological model where we insert the polymorphism. And this has been very interesting because in one particular gene, brain-derived neurotrophin factor, that is absolutely key to learning and development. The phenotype for this mouse is that it's incredibly anxious. And the other interesting component about this mouse's behavior is that it is very hard, once it learns that a cue is associated with negative information, for it to habituate or extinguish, which is very similar to some of the work that we're seeing with regard to those individuals who can't regulate their emotions. So we're beginning to see an association, but until we get larger numbers, we really can't tell, but that's a really important part. Just in terms of your comment about 18-year-olds, I was telling a couple people earlier, I spoke to judges across the nation at Johns Hopkins a couple of weekends ago, and several of us tried to convince them at some level we really need to move out to 25, but there is no line that can be drawn to say when you're an adolescent or an adult. In fact, adolescents do very well, and in fact they're faster than we are in making some very good decisions. It's when they're caught in an emotional situation at the heat of the moment that we have to give them some breaks. And so we're trying to encourage them to move that line depending on the issue.
Let me add something as well. Your question about genetic variation is a very important one because, and there is a now very famous study that was based in New Zealand. It was a longitudinal study, still ongoing, a group in Wisconsin and London, a man named Avshalom Caspi. And two findings that are particularly interesting. One, in the case of vulnerability to depression, they determined the presence of two different alleles, variants of common genes that actually have to do with the serotonin system, which is implicated in depression. And if a person had a particular allele, they were less vulnerable then to bad life events precipitating depression, whereas the other allele, also present in the population, made them more vulnerable. But things had to happen to them. They had to have certain adverse life experiences, sort of the second of the two tolerable stress. But the vulnerability factor was conferred by this particular gene. The experience had to occur as well. The other gene, a set of genes, this had to do with the adverse childhood experiences that I mentioned. And this was another gene that's involved with serotonin metabolism and so on, and so again implicated in depression. But in this case, they were looking at the antisocial personality that develops with early childhood adversity. And again, there was an allele that was protective as opposed to another one that increased vulnerability. But in both cases, there had to be child abuse. If you didn't have this abuse, then you could go through life without any problem. But if there was abuse and you had this particular form of the gene that made you more vulnerable, then you were more likely, the kids were more likely to have an antisocial personality. So it's a beautiful illustration of how genes don't determine everything that happens to us, but the experiences that we may have or may not have also interact with those genes to result in psychopathology later in life.
There's a question 2 rows further back. I will get to the back eventually, don't worry.
Thank you again for your presentations. 2 questions. One, do you have a stress test that you used with the adolescents? Or was this a test that was used as well for adults, or is there a new or special test, diagnostic instrument?
So the measurements that we used in this particular study assessed the level of stress, and they were based on self-report. and they were appropriate for the college-age students that Connor was working with. In the other study that I presented with children, we didn't assess their stress, and we got parent ratings in terms of overall anxiety and their everyday feelings of anxiety.
Is there instruments for adolescents? A stress instrument, a diagnostic instrument?
You're not really making a diagnosis, but there are instruments that have been modified for use in children and adolescents. There are also, I thought what you were really wanting to know is you can, there are stress tests where you actually put the individual in a stressful situation. You have them get up and they have to give like a speech in front of a group, things like that, and you can look at the stress response. We're also getting cortisol so that we can measure the biological form of the stress response too. And that's much higher in the individuals who grew up in the orphanage than in those who did not. But those were typically developing.
Were the adolescents from New York City?
In the first study, yes, indeed, they were.
Okay.
I might add, I mentioned that low self-esteem has been associated with a smaller hippocampus. The hippocampus is known in shutting off the stress response as well as being involved in memory. This was first uncovered with this public speaking challenge in a study in Germany about 10 years ago, putting people up in front of a strange group of people to talk about something personal and measuring their cortisol response. Now, for most people, if you do it once, your cortisol goes up. Probably our cortisol went up this evening. But if you do it repeated times, then you habituate. You don't produce cortisol every time. But there was a subgroup of people who produced more cortisol and they didn't habituate. Every single time they were up in front of it. And these were people who initially appeared to have very low self-esteem, and when one of the investigators moved to McGill University and came upon a brain imaging system that he's become involved with, he imaged the brain and found that they had a smaller hippocampus, which is consistent with their overproducing cortisol, but it's also consistent with their having other more cognitive issues. And of course, this is an ongoing study. It's an initial observation. It needs to be confirmed, but it really brings up something which undoubtedly in children who develop in these chaotic environments, they develop low self-esteem, and they're going to have impaired cognitive function.
I just wanted to add that the study I presented today was just performed at the Sackler Institute, but it replicated smaller studies that we performed in Pittsburgh, which, while I thought I was still in the Northeast Corridor, it's Midwest.
Now, there's some questions coming up at the back. So whilst the microphone's going there, I'm going to ask a question, if I may. I mean, I'm asking it as a parent myself, and maybe other parents in the audience might also be interested in this. Knowing that there can be an imbalance between these two parts of the brain, is there anything we can do about it? I mean, is there anything that you can suggest that would help?
Well, so, I mean, I'm going to turn around and make it an even more interesting question. Not that your question wasn't interesting. Wow, okay.
Are you trying to stress me?
I'm just thinking what my mother would say right now. So, I mean, the question I would be asking is, why would the brain be wired this way, right? Why would we do this to kids? The amygdala is not the only region that is elevated during adolescence. There's this other region, like the amygdala is sitting there trying to learn what is a cue of safety or danger. There's this other region, the accumbens, that's been associated with risk-taking. And they both can be banging. And so really what we think is, over time, what's happened is adolescents can now go on to 40, you know, in terms of being reliant on your parents, not quite being independent. And it used to be that you had to move out of the village to procreate. Now, if you're in a safe village, why would you want to leave that shelter and security? You need something going on that wants you to change things up a little bit. And so we think that this might be part of why the system is changing. And, you know, I don't want to do We want to watch for cases where there is a significant stressor during this period of development, but adolescents really change up our world. I mean, they drive us crazy, but there's a lot to be learned in terms of patience, and also learning to know when to hear about half of it and when not to hear some of it, because many times they do settle. And recognizing that it's this initial response and that most of these adolescents can bring it down, that's something that we need to keep in mind. Might keep us from saying something too quickly.
I think that in another way of answering, I think we don't know yet of ways that we can change the brain, but I think because the brain is a plastic organ and is capable of change, for example, we know that that in something called Cushing's illness, where there's excessive cortisol, there's a smaller hippocampus, people have depressive symptoms, and they have cognitive impairment. If you take away the pituitary tumor that produces the cortisol, over a year or so, the hippocampus gets larger, mood improves, and cognitive function improves. We need to do more studies of this kind, and I ask people who know about, how to change someone's self-esteem. If you also measured their hippocampus, maybe you could make it get larger again. We think that antidepressant treatments, including physical activity, which is a good antidepressant, may actually enlarge the hippocampus, improve the number of neurons. So I think there's hope out there that we can actually find treatments that may make a difference, not just early in life, but even later on.
Good. Now, I think we've got a microphone. There's been 2 questions for a long time in the middle here. Wherever you want to go, just do it.
I think it's very encouraging that you can improve our hippocampus. I think it's great news. I thought we got more and more plastic and less pliable as we got older. But my question is, does this all boil down to the fact that it's nurture versus nature, that nurture is more important than nature?
We can both take that. I think your point was. I don't think that that's what it's saying at all. In fact, I think the CASEY study that Bruce referred to really indicated that we can't think of these as separate, that they interact. And so you actually, there are experiences that you need that can actually turn on and off genes. So that's something to keep in mind. Did you want to add to that?
Well, I think just to repeat what I said, genes, most of the genes that we deal with most of the diseases of modern life, whether it's Alzheimer's, depression, diabetes, so forth, are not death sentences for having the disease. They determine increased vulnerability or decreased vulnerability. But you have to have various experiences in order for these disorders to be manifested. And by the same token, we can probably find out ways of preventing them. And of course, the ultimate hope of maybe combining genetics is to recognize genes that make you vulnerable and then be especially conscious of the people who may need special interventions so that you can ward off the development of a disorder.
Good. Questions up in the middle.
Hello. What would you say the implications are for teenagers that go off to war and then return as teenagers for themselves and also for their families? And what suggestions or ideas do you have given the research that you presented?
That's a very important question.
There's also the child soldiers that we hear about. It's I think an incredible challenge, and it's not just young people. It's people who are in their 30s and even 40s who experience being removed from their families, going to a war zone, being exposed to the horrors of war. And as we all hear, there's not only a problem with head trauma and other injuries, but a problem with post-traumatic stress disorder. And part of it has to do with the experiences, but also part that people have had and bring home. Part of it also has to do with the reintegration with their families and with society, and the fact that we don't do enough to really help that process. And I think that, again, the hope is, because the brain is a malleable organ, that if we develop better strategies for welcoming people back and monitoring them, that we can actually reduce the incidence of some of the psychopathology that we know exists.
Okay, still in the middle there. There's two questioners on the right, then one on the left.
Hi. Have you done any studies on children who are on stimulants such as Ritalin or Concerta and how that might affect their ability to manage stress and their reactivity?
So another area of my research is on risk-taking and also impulsivity. We have not looked at stress factors with this population. We have looked at the effects of stimulants on the brain and how well it actually helps them regulate their behavior. This was part of a large study funded by the NIH. There were 6 different sites. We were following up on a sample that had been examined earlier, but no biological measures had been used, where 3 of the investigators at different institutions were believers in behavioral intervention. 3 of the investigators were interested, or, well, medication was necessary. The result of that trial was 4 severe cases of combined ADHD, hyperactivity, and inattention. The stimulants were the only manipulation that took them above the placebo, which was community help. So we do know that stimulants, particularly the earlier ones, perhaps not so much with Concerta, that they have effects on growth. And so there's more of an effect than just on brain and behavior that we need to keep in mind.
What about anxiety?
Anxiety, to my knowledge, I know of no studies.
I'm only going to be able to take 2 more questions, I think. So I'm sorry about that. But one at the, yes, we got you at the back, and then one more.
I was just wondering, I don't know if you've done any studies on illness early on in life or any stints. For example, I have a premature baby. She spent 4 weeks in the NICU, and how that affects the brain and the growth. I don't know if there's anything, because you did speak about sort of the trauma of losing a parent, but what about the trauma of being very ill when you're very young and how that comes back?
I think that there have been a number of wonderful efforts to make the NICU less stressful and more conducive to these young infants and to their families. And I think there have been some initial studies to suggest that these infants do quite well. Again, there's a lot of individual variability, and it really sort of depends on the complications that ensued. But the nice thing that we found with, the children in the orphanage, really, some of that work was based on even discoveries about hospitalization of children and the lasting effects that it can have. But what we've typically found is if those children are with the family by 6 to 12 months of age, that really you don't see any differences whatsoever. So this early period of time in a hospital hopefully would have a good prognosis given that other complications were resolved.
And one more question, which I think is over here on the left.
Thank you. What are the effects of, and maybe this is a general question, but what are the effects of drug and alcohol with this adolescent population and stress? Does stress bring on that experimentation, or does the experimentation, how does that connect with stress, I guess is my general question.
I'm going to let Bruce take this after me, but Nora Volkow, the director of the National Institute of Drug Abuse, and someone who I collaborated with when she was at Brookhaven Labs before, has pretty much convinced me that if we could lock up our children until they're 21 so they're never exposed to these substances, it would be a really good thing in terms of later abuse and addiction. It's interesting, there's actually a model of substance abuse that shows an imbalance between these actual brain regions, and which comes first is sort of hard to tell, but when I talk about systems that are associated with incentive and risks, imagine if this system is banging as hard as it's ever going to bang, the rush you could get if it's associated with incentives, and how you want to keep getting back to that. But as you develop, you'll never get back to that stage because you have this prefrontal system kind of shutting you down. So you can imagine that would be one pathway to some of these disorders, particularly addiction.
And I think if we look at the adverse childhood experiences study, I mean, it's been very clear that those experiences lead to increased levels of antisocial behavior, substance abuse, sexual precocity, and later on depression, vulnerability to post-traumatic stress disorder. So this would make the extreme case of the hyperactive amygdala and perhaps the underdeveloped prefrontal cortex. Chaos and lack of development of these self-control mechanisms. And then, as I keep emphasizing, there are systemic effects, diabetes, obesity, other aspects of physical illness that also manifest. There's a study from the CASEY, the New Zealand study, that was recently published showing that 20 years after adverse childhood experiences, people have increased levels of inflammatory cytokines, which are the inflammatory system is involved in cardiovascular disease, diabetes, many disorders of modern life, including some evidence that it may be a contributing factor to depression. So you can have these very long-lasting effects of these adverse experiences, which represent an exaggeration of things which happen in the normal population.
Do we have one more question, or can we make a comment?
You can make a comment.
So I just wanted to make one more comment about something that Bruce touched on, and that is the effects of exercise on these brain systems. Some of my favorite neurologists, that's the first thing that they say to their patients if they feel like they're having symptoms of depression. But I particularly worry about this with our teenagers these days when they are so glued to computers, and they're very good at things that we may not be as good at, but the physical exercise, I think, is going to be very important for helping to modulate mood, just based on its effect that it has on the system.
And the social interactions that come from often going out and interacting with other people.
You know, I'm very sorry to have to stop this discussion. It's really interesting, very interesting questions. I should say that we're going to have a reception outside the auditorium, and both Bruce and BJ will be there, so we can continue the discussion there, and any of the questions that couldn't be asked could be asked there. I want to thank you all for coming. As a mark of this inaugural meeting, we have a gift for you, and that gift is a book, The End of Stress as We Know It. Bruce is the author of that book, and we have copies outside of the auditorium. But now I'd like you to join me in thanking two great presenters. I think it was great.